This is a production of Cornell University.
Thank you so much for the introduction. So it's my pleasure to present the research that has been going in my group for the last 3 years. So my lab is focusing on water stress related issues to agriculture. So in here, you've got an overview of all kinds of issues that are pestering the modern agriculture. And most of the issues that are related to water stress, either there is too much of water or too little of water, or the water is salty, are presented by blue dots. So you can see that there are quite a lot of blue dots. And how can the water is becoming a hot commodity in the current climate, especially fresh water. So how can we manage that the agriculture is going to be providing us with food security in a sustainable way? So one of that is diversify our agriculture, learn as much from the species that are already tolerant to those extreme conditions. And here is, don't be fooled by my Photoshop skills. Here is a desert rose that is native to a very dry island of Yemen, Socotra, growing some edible fruits. But then another way that we can ensure to future-proof our agricultural system is to look into the intraspecific variation, like forward genetic studies, species that are already adapted within our agriculture, like tomatoes, wheat, barley, look at all of the land races and all of the cultural important materials, and how can explore the tolerance within those lines to see how can we implement those stress tolerance mechanisms into the germplasm that is being grown on the big scale. And so we're exploring within my lab, we're exploring water stress resilience across multiple species. We're, of course, using Arabidopsis thaliana, which is the crop of knowledge. And we're using the wild tomato, which is the close relative to a cultivated tomato. We're also using peppery bean, which is a close relative to a common bean, a Phaseodons. I'm not really going to talk too much about the peppery bean because of the time constraints. And we're also using cowpea, which is a Vigna. Cowpea has been an incredibly important crop in the US agriculture at the beginning of the last century. But since the introduction of soy, its contribution to the agriculture has diminished. Nevertheless, it is still serving as a very important cover crop. And then before I kind of go and explore all of the things that we're doing, this is kind of the discovery pipeline that we're applying within my lab. So working on all kinds of architectural traits is complicated because you can, especially in the context of natural diversity panels, you cannot really look at the relation between your phenotype of interest and the overall yield, because natural diversity panels are different for many different reasons. So those are not mutant studies. So what we're doing is we're saying we have a trait of interest or a phene of interest. So phene is kind of like an atomic value of a phenotype. Phene to a phenotype is the same relationship as a gene to a genotype. So it's a very, very atomic thing. So rather than how I'm looking today, so this is my phenotype, but the specific phene is the length of my hair or color of my eyes, stuff like that, right? So we're selecting a phene of interest that we think is contributing to the plant performance. We look at what is the plasticity in response to stress. That's kind of the things that we're interested in. Of course, we're looking at the heritability, because if your phene is not heritable, in our specific case, we're not going to find underlying genetic mechanisms. Then we're going into population screening. So we're doing phenotyping. Sometimes it's high throughput, sometimes it's high dedication. And then we're doing forward genetics. Once we've done the forward genetics, we have identified few genes of interest. And then we're doing the validation using reverse genetics, which is making transgenic plants, gain of function, loss of function, and identify their molecular context. And only at this stage are we looking at the relationship with overall plant tolerance to stress and overall relationship to yield, because we're comparing the wild type to our gain or loss of function mutant, and those plants are really differing only hopefully in the phene of interest, in the gene of interest, and not in all kinds of things that are also affecting the yield. So first is the Arabidopsis. So I started with Arabidopsis during my PhD in Amsterdam. And when you're exposing the plants to increasing concentrations of salt stress, they are becoming smaller. My question is, are they also becoming any different? Are they salt stress-induced changes in the plant architecture? So we developed some very simple models to describe the root system architecture as 3 components. This is all published work. We identify that, yes, salt is not only slowing down the development of the roots, but actively changing the architecture of the plant, of the roots. Then, of course, we put it into the GWAS. We screened 350 accessions of Arabidopsis. This population, as you can see, is extremely Eurocentric because people that have collected this Arabidopsis have been making a lot of research in Europe and collecting the plants around Europe. Since then, that was done in 2004. Since then, we have also collected Arabidopsis all over the world, including Africa and Saudi Arabia, where I did my postdoc. So after starting my postdoc, I was a root biologist, and I'm a very proud person to be a root biologist, because as a root biologist, we're looking at the hidden half of the plant, but it's still a half. So after doing all of the study on the salt-induced changes in the root system architecture, we're starting to look at what is the salt-induced changes to root and shoot ratio. And we know that root to shoot ratio is extremely important when it comes to drought. So plants are prioritizing scavenging for water and minimizing the transpiration. So the root to shoot ratio is changing in response to drought. How is it changing in response to salt? And so we thought, I had all of those scanned agar plates for my PhD, might as well reuse the images. So together with Rashid at KAUST, we developed a very simple tool that is basically isolating green pixels, white pixels, and the blue pixels for the root, shoot, and the background. And we quantified semi-manually or semi-automatically, but semi-automatic always means semi-manual. Someone has to do all of this work, and you only realize it when you're doing it. So we quantified all of those pixels. Of course, we have problems with this tool because if the plants are growing into each other, we cannot really separate the roots. So it's not a perfect tool. Anyway, let's cut the chase, go to the results. So in here, we've got the growth rates of root and shoot under control conditions, salt, more salt. So unsurprisingly, the more salt you add, the slower the root is going to grow, and the slower the shoot is going to grow. Not a big surprise over there. But what happens when we look at the root to shoot ratio? So under control conditions, we see that the root to shoot ratio is around 1, which means that the growth of the root and the shoot is fairly well coordinated. As soon as you add salt, we can see that this variation is starting to spread in both directions, which means that the root and the shoot coordination is kind of disturbed. So some of the plants are becoming rootier, some of them are becoming shootier, and we cannot really find a big trend if whether shootier or rootier trend is overall better for salt stress tolerance. So what we did is we threw all of this data into genome-wide association. When you do genome-wide association, this is the kind of results you're getting. So in here on the x-axis, we've got 5 chromosomes of Arabidopsis with all of the positions of the SNPs. And here we've got an association strength. And if your trait, and in our case, shoot over total area of the seedling at high salt stress concentration is associated with any specific genetic loci, you can see that your association strength is going up, right? So in here we've got a couple of SNPs. Then we got a new data set of the SNP being released. So this is the genome-wide association mapping with only 250,000. Here's genome-wide association with 4 million SNPs. So we've got much higher resolution. But also your threshold for significance is going up because we need to do a Bonferroni threshold. So the more things you're testing, the lower p-value you actually are chasing for. So it's not significant, but it was significant before. So let's still have a look at that. So all of the previously significant SNPs are here in red in this gene called DAF247. And it's neighboring all of the other DAF247. It's like DAF247 Valley. What does DAF247 stands for? It stands for Domain of Unknown Function. Thank you very much. So then with all of the increased SNPs, we're mapping, all of the SNPs are mapping to this one DAF247. So we're like, okay, great, this is fantastic, but what does DAF do? So we're looking at available expression data. It goes up under salt stress, but relatively at the very short term, and then it goes down again. It doesn't seem to be super specific to other stresses, like drought or osmotic a little bit, but not too cold or anything else. So what we did is we looked for the mutants, so loss of function mutants, and that's all of the data done by Mariam, who is here in the audience. So she was doing a lot of high dedication phenotyping of root system architecture of plants grown under control conditions, salt and more salt. And what she found is that we see a slower development of the lateral roots of the DUF mutant at the low salt conditions, but we don't see a significant effect under high salt conditions. And we don't see any effect under no stress conditions, which is nice. So we see something is happening with this lateral root development. Okay. So then we say, like, okay, is this effect only specific to agar plates? Because agar plates, although they are native environment for Arabidopsis, they're not exactly native environment for many plants. So we wanted to grow them in the soil. So we're using the system that we have developed at the BTI called the PhenoCage. So you've got Arabidopsis plants growing under the Raspberry Pi camera, and we're making images every 30 minutes, which allows us to track the growth rate. So what Mariam did again is she grew the plants, Columbia and DUF mutants, under control conditions after early stress. So the stress was applied 2 weeks after germination, and late stress, so the stress was applied later on, 3 weeks after germination. And after early stress, we start seeing that the Columbia and the DUF is actually growing much better compared to the Columbia. So the mutants, although it developed less lateral roots, it's performing better under soil conditions with salt stress. We don't see much difference under non-stress conditions again. So we also had a look at, okay, do we see any differences in the shoot ion leakage? So ion leakage is an assay that we're doing to see what is the cell integrity. And if your plant is very healthy, you have very little ion leakage. And salt stress typically tends to induce ion leakage, as you see in here in Colombia. So we see in here that the DUF is actually experiencing less ion leakage. And we also measured the sodium accumulation in the rosettes. And again, DUF, so after salt stress, we're seeing increased accumulation of salt stress, of salt ion, sorry. And then the DUF mutant has a slightly lower accumulation of sodium ions, which is quite interesting. And it's in line with, like, they perform better, therefore they have less damage and they accumulate less sodium. All adds up. Another thing, so kind of going back to the natural variation, what I like to do whenever I find a gene that is associated with a specific trait, we find the gene, but then, like, what is the natural variation that is actually causal to that specific phenotype? Because natural variation is not that there are accessions where the gene is non-functional or dysfunctional, but it could be functional but slightly differently. So what I do is I put all of the SNPs that are within the gene coding body of my gene, and I'm trying to do the haplotype analysis. So I'm, rather than with GWAS, we're doing it per SNP. Every SNP, we're kind of splitting a population in two. Is that specific SNP significantly different from our control group? With haplotype analysis, we're kind of taking multiple SNPs together. So it's kind of like we're making words rather than letters. So a specific SNP combination. And our haplotype 1 is where the Columbia is, our reference genotype. And haplotype 11 was another set of genes. And every single haplotype needs to be represented by at least 3 different accessions. Why 3? Because you cannot make a box plot with less than 3 points. So we found this haplotype 11 was significantly shootier compared to Columbia under salt stress. So we're like, okay, well, let's start cloning those genes and seeing, like, what is actually the variation rather than the SNPs. So once we started cloning it together with Mashael, that was back at KAUST, Mashael was very concerned because all of the sequencing results from the haplotype 11 came up with those huge gaps. And it turns out all of the accessions that were belonging to haplotype 11 had 200 base pairs insertions, which led to a premature stop codon. So DUF247, domain of unknown function, it's still a domain. And we have a predicted transmembrane domain at the end of the protein, and we've got a domain of real unknown function at the very beginning. So in the Columbia, this is kind of the predicted protein structure. In the haplotype 11 or accession VLH1, we've got this kind of structure, but it's predicted. So we still don't really know. Okay, so that kind of led us to think, okay, what is the function of all of those different domains? So multiple people have been involved in cloning of all of the different constructs. So we've got Coyote, we've got Rita, and Mariam finally had all of the constructs in her hands to examine what is the protein localization within the tobacco transient leaf transformation. So we chopped, so this is the sequence of all of the protein. we've got the nuclear localization signal in here, we've got the coiled call domain in here, here we've got the putative calcium binding domain, and here's the transmembrane domain. So we basically split, oh yeah, and here, this Y, this is where the premature stop codon is in our BLH1 accession. So basically what we split is we split the accession for this first domain, so this is our fragment A, this is our fragment B, all the way to transmembrane domain, and this is our fragment C. So the full-length protein, it seems like it's localizing to a plasma membrane. Whenever we look only at the AB domain, so we chop off the transmembrane domain, its localization is really translocated predominantly in the cell nucleus. However, if we only look at the domain A or domain B, we've got still much more nuclear localization, but we see also a cytoplasmic localization in those things as well. So it seems like the transmembrane domain is really kind of holding the protein at the plasma membrane, and once that's chopped off by whatever, something, it's translocating into the nucleus and possibly binding it. We have done a yeast to hybrid screen, and the only protein that came up is another DAF. So that's fine. And we also have looked at what are the proteins that might be possibly regulating the DAF expression upstream. And Mariam has digged deeply into the literature, and she found that Verkey 75 is possibly regulating it. We have done some preliminary assays with whether we see a DAF expression in the Verkey mutant. We don't. It's totally wiped out, which is nice. But we're doing more assays to kind of confirm the molecular context of the DAF so far. Another thing that we have done is looked at where the DAF promoter is actually expressed. So the DAF promoter, so here we've got seedling grown under control conditions, salt stress, and we see that it's really expressed predominantly in the STEL cells. So STEL is where all of the region around the vasculature, right? And Mariam also looked at the expression in the older leaf tissues. We don't see it being, we only see a slight expression in the young leaves, but then the older the leaves are getting, the lower the DAF expression is, and we don't see any expression in the pollen, which is interesting because some of the other DAF247 genes were involved in like autoimmunity, autosterility issues. We also have looked at quantified the signal of the gas throughout the root in response to salt, and we're seeing that there are some highlights whenever the lateral roots are coming up. And we're seeing that when we quantify all of these gas signals, we see that the signal on their salt stress is higher, significantly higher compared to the control conditions. So the promoter activity is enhanced by salt stress. And this localization in the STEL, so STEL is the region where a lot of signals are coming for the suberization of the root. Suberization in terms of salt stress is extremely important because it forms this hydrophobic layer, which prevents water loss, but also ion entry into the root, which is like, well, in all of those phenotypes that we're seeing, like the DAF mutants are having lower accumulation of salt, lower cellular damage. It all makes sense. So let's have a look if we see changes in the root suberization. So here we've got Colombia under control conditions and salt stress, and you can see that the salt by itself is already inducing the root suberization. And then the DAF mutants, what we see in here on those semi-quantitative pictures from the confocal microscopy that Mariam did, is we're seeing that the DAF overall is having a slightly higher intensity when it comes to the suberin staining. We also quantified it in collaboration with George Drakakaki from the University of Davis. And we found that some of the components of suberin, like total fatty acid, fatty alcohols, are induced in the DAF mutants compared to the Colombia, especially in salt, but some of them are also induced under control conditions, which is fine. So the DAF story thus far, we know that the DAF247, our specific DAF247, is expressed during early vegetative stages of development. We know that the full-length DAF is residing in the plasma membrane, and once we chop off the transmembrane domain, it relocates to nucleus. It does something there. We don't know yet what. DAF seems to be limiting salt tolerance. So if you don't have the DAF, you're better off when you're experiencing salt stress. And it's probably through negative regulation of apoplastic root barriers. And right now we're developing the RNAi and overexpression lines in Arabidopsis that are targeting multiple DAFs to evaluate like whether we knock out our DAF only or multiple DAFs at the same time. Do we see a compound effect? And I told you that we identified one interactor of DAF through yeast to hybrid, but it didn't provide us much clarity. Okay, so moving on. We're also investigating other plants, as I told you. So Arabidopsis is only one part of the lab. We're investigating wild tomato, which is native to South America in here, which spans a range of different climates, including Galapagos Islands. So those are together with Chismania and Galapagense, Pimpinellifolium on one of those tomatoes that are, described as growing into the sea breeze and being extremely salt tolerant. Peppery bean is another species that we started to work on, which is native to southwest of US and northern Mexico. It has been used in native agriculture for quite some time right now, and we're very thankful to kind of explore more of this germplasm. And another species of interest is cowpea, which is native to sub-Saharan Africa, and it has been grown by subsistence farmers under non-irrigated conditions, which is relying on whatever is falling from the sky, which is sometimes not so much. So Pimpinellifolium is the closest relative to cultivated tomato, and it has been previously used to integrate some of the traits into a cultivated tomato, like the stemless gene or resilience to many viruses and biotic stresses, as it's native to dry and coastal regions of Peru, Ecuador, and Galapagos. It has high salinity, drought, and heat tolerance. We've got a reference genome sequence available that has been done by BTI, by FaceLab. And we have sequenced 230 accessions at KAUST, and we phenotyped it in the field conditions and in the plant accelerator, so under controlled greenhouse conditions in Australia. The paper has just been accepted at the bioRxiv, so you can go and check out if you're interested in more details. So me being myself, I of course screened the whole population for the root system architecture. So here we're only growing one plant per plate in here because the tomato has tremendous root system architecture. We're growing them under control conditions and salt. And of course, later on, it follows with high dedication root phenotyping. And it was done during the pandemic by myself and Haley, who is a technician in my lab. So here are the results, basically. So we're looking at the main root growth, the lateral root increase, and the lateral root growth under control, blue, and salt stress conditions. And you can see that main root growth is actually not that much affected in this Solanum pimpinellifolium population, but the number of lateral root is, and the lateral root length also not so much if you look at the overall population levels. However, if you compare individual plants, how they are reacting under control and salt stress, we can start seeing that there is definitely a genotype specificity effect over there for all of the traits, almost all of the traits. For the average lateral root length, not so much. However, if we look at the relative performance of the plants, so are there different strategies for different accessions in terms of salt tolerance? We see that there is also different patterns. So for example, we see plants that are not really phased by salt in terms of main root growth, but they are really suffering when it comes to lateral root development. And we see, of course, other opposite patterns, like they are very much sensitive in their main root growth and not so much when it comes to lateral root development. So kind of based on that, we selected one accession of Pimpinellifolium, which is M248, which develops a high number of lateral roots, but those lateral roots their length is very finite. And we also started screening a cultivated population of tomato, where we've got exactly the opposite strategy. So both of those plants are salt tolerant, but one keeps on popping up new lateral roots. The other one doesn't really pop up new lateral roots, but it elongates whatever it has. So we're like, those are really interesting opposing strategies. Is there anyone better? No, there's not. They're just as tolerant. If we cross them, so Mariam has crossed, again, Mariam is the hero of the lab. So if she crossed both of those lines, with F1, we see that the babies are inheriting both of the positive traits, which means that there is no negative trade-off, which is great. So right now, we did an F2 screen, and we're doing a bulk segregant analysis. So we've got the plants that are superheroes. So they keep on popping new lateral roots and elongating them. We've got the super losers, which are having problems with elongation and popping of the new roots. And we've got the intermediates, which are similar to both of the parents. So right now, we just got back the sequences for the bulk segregant analysis, and hopefully we can put that bulk segregant analysis on top of our genome-wide association that we've got for the lateral roots and see which of the genetic loci are contributing to the lateral root popping and lateral root elongation under salt stress. And hopefully they're different loci. However, so we're not finished yet with tomato story, because if you spend a lot of time with your data, what I did during a pandemic, you start seeing things, and sometimes those things are real, sometimes they are not real. So when I started drawing all of those roots, there are specific patterns that kept on popping up. And I started seeing like all of the roots that are looking exactly like an upside down Christmas tree, something like a telephone pole. So you've got the straight root and all of the lateral roots are having very finite length, but they're all getting out. Then we've got the little droopy telephone pole, and then we've got the broomstick. So majority of your lateral roots are popping up at the root base. I'm like, this is interesting, but how do I, like, you see something and like, how do we get it quantified in actual stuff that we count, like lateral root number, average lateral root size, branch zone size, all of those things, like canonical traits that we're normally using for describing roots. So I was like, okay, maybe I can make some kind of mind map because I see it, but I don't, like, I always have one comparison between my Christmas tree, telephone pole, droopy telephone pole, and broomstick. There's always one non-significant comparison. So how do I get to describe what I see in here with numbers? And then I started talking to mathematicians, and Arjun Chandrasekhar, who is right now at Southwestern University, he was like, we should use Pareto front. Because if we use the Pareto front location, we can see beautiful differences between all of those different ideotypes. I'm like, great, what is Pareto front? So Pareto front is a metric that people are using for evaluation of infrastructures in the cities. So what you've got in here, so here is a subway system of Amsterdam, which has recently undergone an expansion that costed the whole city millions of dollars because they tried to dig tunnels under very old buildings. So every single time that you're building a new branch of your network is extremely expensive. Similarly to the plant, every single time you're making a new route, you need to invest some effort, some building blocks into that. So and you can be fast or cheap. You cannot be cheap and fast. That doesn't exist. So if you look at the root system architecture, we've got main route as a kind of highway. It has to exist there. And you've got lateral roots that are performing the most of the radial transport of nutrients and water. So radial transport is we're absorbing water from outside of the soil into the vasculature, and that goes up to support the growth of the shoot, right? So there are multiple ways of connecting those root tips to the main root highway. So we can, if we abstract that, so we've got in here the root as we see it. Let's say we only know the position of the lateral root tips and the main root highway. So we can connect it in a very fast way, we can connect it in a very cheap way, or we can connect it in the intermediate way. So that means that you don't really have one solution that is ideal. You've got a whole range of ideal solutions. Right? So you've got the front of optimal solutions, and we want to know how far or where on this front of optimal solutions we are. So that kind of determines our Christmas tree, broomstick, or telephone pole, right? And then, so we've got here the front of optimal solution. So the alpha is your location on the front. And then the other thing is you can also say, what is the distance to my front of optimal solutions, right? Because the plants are having all of the other things that they need to be doing. So when we look at the distance from the Pareto front, the plants that are growing under salt stress, which are the red lines, and the plants that are growing under control conditions, which are blue lines, you can see that the red lines are significantly smaller. So they have smaller distance from the optimal front of solutions, which means that the plants are growing much more efficient under stress. compared to whenever they are growing under control conditions when there's no stress, which I can relate to. When I'm stressed, I'm extremely efficient. If I'm not stressed, I'm going around and doing all kinds of things. When we looked at the Pareto front location, so this is the alpha, so it's whether you're a broomstick or a telephone pole, we see that the data is much more noisier, and that is mainly because the system, the Pareto front location system, is not exactly developed for dynamic systems, because infrastructure in the city, it is a little bit dynamic, but not so much. So, and the other point before I go further is we put all of this data into the GWAS. The heritability is surprisingly high, so that's a great thing. And we are finding some of the associated genes. So in here we've got tomato with 12 chromosomes. That's why we've got a little bit more colors over here. But we're not following any of those genes just yet because we need to fix our model first. And we recently got an NSF mathematical biology grant to do that. So there are a couple of things. So first of all, we need to expand the model to accommodate lateral root gravitropism. So right now, all of the lateral root tips are connected with the straight line to the main root. We're not having like this quadratic or exponential shape integrated in our model. The other thing is that the lateral root conductivity in terms of water and nutrients is very different from the main root conductivity. So if you look at the cross sections that I did for the grant, is you see here the cross-section of the main root. The blue bits are where the xylem vessels are, and here's the cross-section of the lateral roots. So you can see that the blue bits are much bigger in the main root compared to the lateral root. So you've got a significantly different kind of pipe that you have to squeeze through. The bigger your pipes, the faster you can go. But we're not integrating that in the model just yet. So we need to make sure that we're integrating that across the developmental axis of the root, but also differentiating between the lateral roots and the main root. And then we also found that, together with Mariam again, we found that when we are applying hormones to our roots, we can significantly alter the Pareto front location. So in here, we're changing this broomstick to a telephone pole by adding auxin. And additionally, when we're adding auxin, it is significantly increasing sodium accumulation in our roots. So seemingly, the telephone pole might not be the best strategy when you want to limit the ion accumulation in your root and in your shoot. So we're figuring that one out as well. And right now, so Trent is an undergrad student that is working in my lab, and he has been working meticulously through the summer, making the cross sections of different developmental zones of the main root. And right now he's going to do all of the different developmental zones of the lateral roots as well. And once we've got that, we can put that into updated model, and all of the phenotyping is done. So that's great. So we can recalculate our Pareto front optimization and put that in again into the genome-wide association and identify underlying genes, which is really nice. So let's switch to another. I'm fine. Oh, okay. Let's switch to another crop that we started to work on is cowpea. So cowpea, as I already told you, is native to sub-Saharan Africa. You can see in here, the natural diversity panel for cowpea has already been developed by Maria Munoz Amatrian and a lot of people from the Nigeria Institute for Tropical Agriculture, IITA. And they have already genotyped the entire diversity panel for the global diversity panel for cowpea. We also are having collaboration with Ahmadu Bello University in Nigeria to genotype a local Nigerian diversity panel. And having global and local diversity panels helps because you've got a different interference of population structure in your forward genetic studies. So we've got a reference genome, we've got a beautiful diversity panel. So let's phenotype them. So phenotyping cowpea is much more difficult than phenotyping Arabidopsis. First of all, Arabidopsis grows like a pancake. Cowpea is growing more like a human. So we need to develop tools to phenotype plants with complex architecture. So we went to Lowe's, we got some wood together, and we constructed this pheno cage. So basically, it's a set of wooden bits. And here you've got the rotating platform where you're putting your plant on. The plant keeps on rotating. In here, we've got a little Raspberry Pi camera. So camera keeps on flashing on the plant, and we've got 7 different side views of the plant. So this is how it looks like. as the cowpea plant is growing. And we're isolating all of the pixels that are belonging to the cowpea from the background using a plant CV. So it is very automated because for every single experiment, we've got around 10,000 of images. So you don't want to be doing that in a high dedication way. We looked at the correlation between the shoot area that we're getting in pixels and the fresh weight of the final day of the experiment. We've got a beautiful high correlation for cowpea as well as for tepary bean, because tepary bean is also growing weird, like in the 3D. For many different legumes, because they've got a different growth style, so not all of them are standing by themselves. You've got plants that are prostrate or climbing, so we need some kind of structures to support them. So what we did is we 3D printed support structures, those trellises that you can see here from the transparent filament, and they basically disappear in the pictures. So we can get quite a lot of precise data for that. And if you want to know more, again, the preprint is available on the bioRxiv. So send me an email if you want more details on that. Another thing that we also did is we are also interested in like how much water is a plant using every single day, and we're watering, especially in the drought stress, we're watering the plants to a specific target weight. So watering them, before and after watering, all of the data is collected, and that allows us to estimate the evapotranspiration of the plant. So here, Olga Khmelninsky developed this Arduino-based system that is connected to a pump and a scale, and this is how it looks in real life. And we can track the evapotranspiration under control conditions and drought stress. You can see that the plants are evapotranspiring much less. This is quick and dirty data, of course. It's much less precise than all of the Likert or parameters and stuff like that. But it's still a data that we've got for free. So we screened all of the 368 accessions of cowpea. We see that under stress conditions, they are growing slower, not surprisingly. And we're seeing that they are evapotranspiring also much less controlled compared to the control conditions. We also measured some of the photosynthetic traits using the handheld device, PhotosynQ. So we measured the quantum yield, the leaf temperature, and put all of that into the genome-wide association. So in here, we've got the genome-wide association plot. So we've got 11 chromosomes in cowpea. We found 6, I think. 6 loci. Yes, 6 loci for growth rate, cumulative evapotranspiration, and non-photochemical quenching. We also looked, so this is the association strength. This is the predicted effect size graphs that we're using as well. So it's not only important that your specific SNP is significantly associated with your trait, but if you predictively have a gain or loss of function, is it going to have an effect that you can care about at all? So this is what those graphs are telling us about. So if our specific SNPs are kind of sticking out of the general background noise, it means that, yes, this specific association might be worth exploring. So right now, making transgenic plants in cowpea is extremely difficult because they are not very easy to transform. So what we did is we looked at homologous genes in Arabidopsis. We ordered all of the tDNA insertion lines that were available, and we screened it in our different system that I showed you earlier. We see, so we've got a couple of genes that I'm not going to go into detail. They're all, again, described in the bioRxiv paper. But for most of the genes that we found in Arabidopsis, the knockout or the tDNA insertion is growing much better than Colombia under drought stress. And we're seeing for some of them also much better growth under control conditions. So it's really, we're targeting the vigor of the plant rather than drought-specific responses, which is something that we don't mind, really. One last thing that I wanted to tell you about, because this is extremely exciting thing that we recently started to explore. So in collaboration with Gaurav Mogi, we explored what are the stress-induced changes in the leaf metabolome of cowpea. So we harvested the samples at 3 days, 7 days, and 14 days after drought imposition in many different accessions, or 3 different accessions of cowpea. So you can see in here that after 3 days, we don't really see much of the changes in the metabolome, but as the time goes on, the metabolome is becoming more and more different. So those are the PCA plots, so just general trends. So the longer you experience the stress, the more different the plants are. Okay, and then we of course looked at specific metabolites. such as prolines and stuff, we saw all of those changes as we predicted. But one of the metabolites that was not very obvious was N-acetyltryptophan. N-acetyltryptophan is an acetylated form of tryptophan. That's all I know. The only thing that I could find in the literature is that it's enhancing the germination of one of the orobranchi, which is a striga plant, I think. So one of the parasitic plants that is forming tremendous losses in yield in Africa. But we haven't really had any context for the N-acetyltryptophan in the stress, like abiotic stress biology research. So we saw that this accession UCR799 is showing tremendous increase in NAT, whereas other accessions less so. When we look at comparison of performance of those 5 of those accessions, so in here we've got UCR799 growing under drought stress, we see that this accession is actually performing worse, much worse compared to Suvita. And we see in here, so that the growth trends are exactly the opposite, right? So the more NAT you seem to accumulate, the lower you grow. So we ordered some of the NAT. It was pretty cheap, so that's great. We put many different concentrations. That's the work that was done this summer by an RAU undergrad, Jade Milton. So what they did is tested first different concentrations of NAT on Arabidopsis on the plates. And we see that lower concentrations are enhancing the root growth, whereas higher concentrations are decreasing the root growth, which is fine. Then we're like, okay, what happens if we combine this NAT with osmotic stress? So we use the PEG 8000, and the more osmotic stress you add, the slower your root growth. But in combination with NAT, the root seems to be performing slightly less, but not by much. We, of course, compared also the effect of the NAT to control conditions and also to tryptophan itself and auxin at exactly the same concentration. So those are the results that I just got like earlier this week. So I didn't have time to quantify it yet, but you can see clearly that tryptophan and N-acetyltryptophan are having tremendously different effects from each other. So this N-acetylation is really having a huge, huge difference. And I also didn't know that tryptophan is causing this kind of effects on the root system architecture. So what do you know? We also looked at the effect of N-acetyltryptophan, what happens when we add the N-acetyltryptophan to the plants growing in soil. Before I tried spraying them because we found N-acetyltryptophan in the leaves. When I spray them, nothing happens. When we add them to the soil, what we're starting to see is this interveinal chlorosis, which is really interesting because normally chlorosis is occurring between the veins, but not from within the veins. So there's maybe some kind of toxicity or maybe some kind of nutrient deficiency going on. When we measure the chlorophyll fluorescence, we see that FV/FM, which is the light-adapted quantum yield, in the old leaves, we're seeing that NAT is significantly decreasing the chlorophyll fluorescence or the quantum yield of the leaves, which is something that you would expect. And we're also seeing that chlorophyll content is decreased by the NAT. When we measured the specific accumulation of elements in collaboration with Miguel, so what Jade did is they cut the leaf in the edge, the veins and the interveinal regions, and we measured all of the different elements that we could measure. The only elements that we saw significant differences were nickel. So the NAT is causing increase in the nickel, but only in the edge, not in the vein, not in the interveinal region. We see an increase in the phosphorus content. We see a tremendous increase in the sulfur content, and we see a tiny increase in the copper content. So if anyone has any idea what this could be, please let me know. So then that's kind of wrapping up my talk. So take-home messages is that exploring the responses to stress with focus on plant architecture rather than just general plant size might lead you to discovery of new components of resilience, for example, DAF247. Studying stress resilience plants like cowpea or wild tomato can provide new strategies of quantifying the responses to stress, like the Pareto front optimality. that we observed. And dipping into untargeted metabolomics can provide you with a lot of headache, but also potential new players in stress resilience, like N-acetyltryptophan. And with that, I would like to thank you for your attention, for the opportunity to present in front of such a distinguished crowd of young and more experienced researchers. And of course, all of my collaborators, all of the groups that I have been a part of and I'm still a part of in here. Thank you so much. There's one.
Very, very fun. So I have I saw all plans selected for default stress for all my dipods. Have I actually or selecting dipods?
So my focus on dicots rather than monocots is, I don't know. The legumes. I think the monocots, there's quite a lot of interest in it because there is a lot of agricultural interest in it. I think the monocots, the dicots are usually much more high value fruits, much more richer in different micronutrients, macronutrients, and that's the main reason to kind of like help the communities that have not had that much attention just yet, and trying to understand it on the fundamental levels as well as develop resources for the underprivileged communities. Yes.
Probably there may be like something different.
Yes.
Features that like
how yes.
They resistant to the draw strategy.
Yes, they've got a very different root types because the monocots have crown roots and they've got two different types of lateral roots. I don't know how many different lateral roots are in the dicots, but it's all worth exploring to kind of like how far can we translate the findings that we're doing in dicots to other species as well. Yes.
Thank you. I agree. So I have an observation that you could respond to this applies to monocot as well as the dicot system. There's some data showing that the anatomical differences in different orders of prime roots significantly correlate with whether the root order is primarily requiring nutrients or transport.
Absolutely.
And you did show some anatomical. So the correlations have to do with the growing size of the seal versus the cortex.
Yes, so we've got collaboration with the plant modelers, root modelers. So Guillaume Lobé is one of our collaborators on the NSF brand. And the thing is that all of the root models for the radial versus axial transport has been developed on the monocots, right? So there's tremendous. No, okay, all right. I'm being okay. Anyway, but so they have developed the water transport models to incorporate the secondary root growth because that's not happening in dicots. So you've got an increased surface of the root as the secondary growth is occurring. Yes.
There is
no. Absolutely,
so
you're right.
Well, so-called juvenile.
Yes. Absolutely.
I think that's fertile ground.
Absolutely. Yeah. So we're trying to accumulate as much of the anatomical data to kind of implement that. And then once we've got the anatomical data, we can kind of try to explore and kind of confirm the models in different developmental stages as well.
You're finding that you get different kinds of information if you go with the raw morphometrics versus the normal types of data.
Absolutely.
So you should look at both.
Yes. Thank you so much. Yeah.
I just have a question about your adopt 247 that you were talking about. So you said it has a transmembrane domain and that might have a calcium binding domain. When you did your yeast 2 hybrid,
yeah,
were you expecting to pull out CBL, CNK networks because those are already implicated in
Yeah, so we did a yeast to hybrid was done with the protein structure without a transmembrane domain, just to make it more soluble.
Oh, okay.
So it might be biased and we might be missing quite a lot of things there. But yeah, we would be expecting some kind of calcium, but it's also like a predicted calcium binding domain. So we haven't validated that it's actually binding calcium at all. So that's, it's very, very, very hypothetical at this stage. Yeah.
Similar to the question, do you think it could work as a channel protein? It's probably not structural, but also not a factor. Do you think it would work by itself as kind of like a channel, or?
I don't think the channel, it might be, I think that the transmembrane domain, because the transmembrane domain is not very specific. It doesn't seem like it's accumulating in a very specific like subdomain of the plasma membrane. So this lack of specificity, I think it's a part of the protein maturation rather than being and acting as a part of the channel. And also like the channel, I would expect much more transmembrane domain if it's a channel or some kind of supportive interactions. Yes.
It seems that the findings you showed us, particularly for the DOF247, were with sodium chloride stress.
Yes.
And I'm wondering if that's a sodium effect or is it a osmotic or water potential?
We don't, well, did we test the osmotic effect? I don't think we did. We did not test the osmotic effect. So we didn't test the calcium, the potassium chloride, we didn't test the pH just yet. Just because the effect of the DOF is so small relatively on the system architecture. So we need to do quite a lot of replicates in order to get significant results.
You tried mannitol?
We have not, but that's definitely in the pipeline. Thank you very much for that suggestion. But what I wanted to do is have more alleles for the DOF, because right now all of our findings are based on one sense of the DOF, which is very dangerous. I realize that.
Well, with that, I'd like to thank Magda again for not only an excellent seminar, but a delightful one.
This has been a production of Cornell University, on the web at cornell.edu.